So our next speaker is Nathan Treff. Nathan is the CSO at Genomic Prediction and is also an associate professor of obstetric gynecology and reproductive science at Rutgers University.
Thank you. I'd like to thank Rita for inviting me to this meeting and to the organizers. I've met a lot of nice people, with the exception of the bioethicist from Belgium. Just kidding. I've been asked to discuss what we should do with mosaic embryos, and of course I'm going to take a little bit different angle to this question than you might expect. First of all, I'd like to disclose that I am a co-founder and chief science officer at a new company called Genomic Prediction, where we offer testing materials and services for predicting aneuploidy, single gene disorders, and polygenic disorders for the first time. I'm also on the Speakers Bureau for EMD Serono. Okay, so one of the things I'd like to point out is that aneuploidy is a very common phenomenon, and if you consider the frequency that it is observed in human embryos, it's actually the most common genetic abnormality in humans. And so I think this point makes the idea of not giving patients information on the evidence for its use for PGS testing actually unethical. It's like selling a car to somebody without airbags. And you know, they may not actually ever need an airbag. Most of us haven't been in a car accident, but I think we should at least know that this is an option that you can have when proceeding with IVF. But I'm not here to talk about why I think PGS is a valid technology, particularly when you use validated methods. I am going to talk about some of the limitations associated with currently available methods of testing. And with regard to mosaicism, I think there's a common misconception that mosaicism is very frequent. The first sentence in this paper suggests that most human embryos are mosaics of euploid and aneuploid cells, and this is just not true. There's no reference, and I'm going to show you some data to demonstrate that this is not the case. We've known for a long time that the majority of aneuploidy found in products of conception originates from maternal meiosis, which means that you'll have uniform aneuploidy within all of the cells in the embryo, rather than mosaicism. You can see some of the origins of aneuploidy shown here. And to summarize that data, you see again that the majority is originating from meiotic errors in the oocyte, again leading to uniform aneuploidy, not mosaic aneuploidy in the majority of cases. Of course, that's in products of conception, and you might think that it may be different in the human embryo, the pre-implantation embryo, and there are several studies, including one that we've presented, evaluating the origin of aneuploidy. In this case, we looked at sequential biopsies of the second polar body and the embryo itself. This is microarray data where you see the copy number for each of the chromosomes. And in the first polar body, you can see a loss of chromosome 14, reciprocal gain in the second polar body of that chromosome, and then a normal status in the resulting embryo. And you can see a variety of other abnormalities that do actually appear within the embryo. And because we see those in the polar bodies, we know they originate from maternal meiosis. And therefore, you expect uniform aneuploidy of those chromosomes in the embryo. There are other situations where you don't observe the aneuploidies in the polar bodies and only see those present in the embryo. And this can therefore originate from mitotic error or a paternal meiotic error. And you can use the SNP genotyping to determine whether or not the error came from the paternal genome or not. And that helps you determine whether or not this is a mitotic error, such that you can sort of distinguish between the origins of aneuploidy in these studies sequentially biopsying the embryo. And again, we see a similar trend in that the majority of aneuploidy in these samples originated from maternal meiosis, again suggesting that we expect most aneuploidy to be uniform and not mosaic. And so of course, there's been randomized controlled trials, systematic evaluation of these technologies, both preclinical and clinical validation, but some groups have reported anecdotal evidence to suggest that it's not predictive of outcome, and you can transfer aneuploid embryos and have a healthy baby. I'm going to talk a little bit about this, which I now refer to as the Gleicher Misconceptions, and many of you may have heard Dr. Gleicher lecture, and he has very nice arguments, one of which has to do with a mathematical model, which I'll talk about, suggesting that trophectoderm biopsy is incapable of reliably predicting aneuploidy in the embryo or determining whether or not it should be transferred or not. He's also cited data from mouse models and, as I mentioned, anecdotal evidence that PGS is not predictive of reproductive potential. And a mathematical model is nice. It's probably one of the things that I don't do. I actually use experimental data. But in this situation, the study involved considering an embryo that has 300 trophectoderm cells. And I would be surprised if Dr. Gleicher's ever produced a blastocyst with 300 trophectoderm cells. Many of us probably haven't either. And I think part of this overestimate of the number is because the quantitation was done on embryos developed past day 5, so this is probably not an accurate estimate of the number of trophectoderm cells. And this, of course, will overestimate the sampling error that you might obtain by removing a few cells from the trophectoderm tissue. And, of course, there are 3 randomized trials showing benefit, which suggests we may not actually need a mathematical model. We can use level 1 evidence of whether or not PGS actually works. And many people argue, well, and I think we saw the question earlier that this may not be applicable to all patients, and I don't think that's a limitation. It just means that we need to be able to better define which patients will actually benefit from incorporating PGS. But there are multiple studies favoring the use of PGS in this situation, looking at the clinical implantation rate. And of course, this is after the first transfer. So if you want to get your patient pregnant in the first attempt, incorporating PGS will increase your success rate. We also have evidence from what we refer to as a non-selection study. This is where embryos are biopsied, but the information from those biopsies is not used to select the embryo. They're selected based on morphology, which is essentially what you're doing when not incorporating PGS, but then we can ask whether or not the PGS result would have actually predicted the actual clinical outcome. And you can see how many embryos were transferred in this situation, which is far greater than the data reported by Gleicher. And in this study, we found 0% sustained implantation for embryos predicted to have aneuploidy, and then a high success rate for those embryos predicted to have euploid chromosome constitution. I think it's pretty clear that the mouse model is irrelevant to human aneuploidy. They have a much different surveillance system to prevent the development of an aneuploid embryo. So this may not be a useful way to evaluate whether or not aneuploidy screening in humans is an effective approach. And also, it's important to understand that this type of technology may not be applicable to programs that have relatively low success rates to begin with. If you're not making blastocysts routinely, then you may not want to use aneuploidy screening until you actually have obtained those high success rates of actually making blastocysts routinely. And this is just an example. You can see the success rates on the national average for the US in 2015, and then Gleicher's rates in orange. And of course, you can see in green the success rate he had with transferring aneuploid embryos. And maybe he stumbled on something that would improve his success rates by actually selecting for aneuploidy. The real issue, and unfortunately, governments have listened to some of these anecdotal-based evidence to make decisions about whether or not patients should have access to this type of test. And Hungary is one of those places where originally PGS was allowed, but then because of recent arguments, it's no longer available to patients. And you can see in this study the increase in miscarriage rate associated with not incorporating aneuploidy screening. And I think this is, again, an unethical approach to evaluating the available evidence, educating patients on whether or not it may or may not be useful, and at least giving them the option of utilizing validated technology for aneuploidy screening. So why are estimates of mosaicism so high? This is an example of a report suggesting 70% mosaicism in 14 normally developing embryos. And the results are displayed here, where you can see color coding for the karyotype in individual blastomeres from cleavage stage embryos. And as I mentioned, the majority of human aneuploidy originates from maternal meiosis. So you would expect at least one embryo to have uniform aneuploidy of all chromosomes. And in fact, none of the embryos in this study had uniform aneuploidy. And so what I'd like you to consider is that some of these technologies do have a false positive rate, which we need to consider. And it has a different impact on different populations of patients. If you have a test with a 10% false positive rate and you apply it to patients that have a baseline 20% aneuploidy rate, the predictive value and the actual clinical value to those patients is much lower than if you have a baseline aneuploidy rate of 50% or greater, where a 10% false positive rate is a smaller percentage of the total of the embryos predicted to have aneuploidy. And this is also an example of why doing randomized controlled trials on patients under 35, it will be more difficult and require a significant sample size to overcome issues such as this. Another example of over-interpreting results and essentially failing to acknowledge that there is a false positive rate with these tests is shown here, where they studied multiple embryos at the cleavage stage and found a variety of abnormalities for which they suggested underwent self-correction, and normal karyotype was obtained 2 days later. And I think, again, the problem here is failing to acknowledge that many of these are likely due to false positives in the technology being used. We actually did a study comparing performance of two platforms. One was ArrayCGH, so we took embryos diagnosed as aneuploidy by ArrayCGH, rebiopsied, blinded, and then performed analysis with qPCR and a second biopsy. We then looked at cases where the two results did not agree using a third test. So based on preponderance of evidence, we could establish which of the two methods gave an error. And here you can see a 9% false positive rate with ArrayCGH. And if we go back to the study where they used ArrayCGH on single cells, you would expect about 47% mosaicism just due to the false positives introduced by the technology. Another problem, it's not just using technologies that have low accuracy, but also how we classify mosaicism in the embryo. And there are a variety of types of evidence for determining whether or not an embryo is truly mosaic. The strongest evidence for mosaicism is if you were to take multiple biopsies and find a reciprocal error. So in this example, we have a monosomy of chromosome 15 in one biopsy and a trisomy of chromosome 15 in a second biopsy. And this indicates that there was a mitotic non-disjunction event, and it's very unlikely that you would see these results due to false positives or errors in the technology, given that they found an error for the same chromosome and that they're reciprocal in nature. So for example, one daughter cell obtained an extra chromosome, and the other daughter cell was missing that chromosome. And this is probably the strongest evidence for an embryo actually being mosaic. And then you go from left to right, you can see single cells with reciprocal aneuploidy would be a high level of evidence. Two biopsies with the same abnormality, either multiple cells or single cells, two different abnormalities. All the way to the right, which is the weakest strength of evidence for an embryo actually being mosaic, but in fact is what is being used clinically to predict mosaicism within the embryo. And this is something that involves looking at intermediate copy number changes. So instead of seeing, for example, a full trisomy where you would expect 3 copies, you might see a copy number in between 2 and 3, which would be something like 2.4, 2.5. And those types of data are being used to predict mosaicism in the embryo. And I would suggest that this is actually the weakest strength of evidence for whether or not mosaicism exists in the embryo. And part of the reason behind that belief is that there are many reasons why you might see these intermediate copy number changes, and not actually due to mosaicism, but other phenomena. When you actually use a high stringency for predicting mosaicism in the embryo, so looking at multiple biopsies, the frequency is actually relatively low. 6% or less of embryos possess mosaicism based on higher strength of evidence for it actually existing. And of course, no matter what you do, when you just take one biopsy from the embryo, you're going to have a sampling error. You cannot predict accurately mosaicism from a single biopsy by definition. The idea is that if we remove multiple cells from the embryo, it's possible that some of those cells will have aneuploidy and some will have euploidy, and we might be able to detect these intermediate copy number changes and successfully detect mosaicism within the biopsy, and then from that suggest that the remaining embryo may be mosaic. And this is an interesting idea and it has potential, but what's missing from a lot of the methods that are actually used clinically is some kind of evaluation of positive controls. And I think this lack of utilization of positive controls for development of tests, particularly in aneuploidy screening, is troublesome. Obviously, when we're doing single gene disorder testing, we evaluate the validity of the test on the patient before applying it to embryos to make sure that it works, and we typically use positive controls. The same principles are true for aneuploidy. You can obtain cells, for example, in this situation, known to possess trisomy 18, and these were male, and we can mix them together with normal female cells at different ratios. Now we can look at these samples that have known mixture levels, so positive controls for certain levels of mosaicism, and ask whether or not the test can successfully detect aneuploidy in these samples. And what we've done is to test a variety of methodologies. Quantitative real-time PCR is shown in black, so you can see, for example, when 50% of the cells have aneuploidy, we can actually predict aneuploidy in those samples roughly 40% of the time. And so that sensitivity increases as you increase the amount of aneuploidy in the sample. We also evaluated a commonly used methodology, which is next-generation sequencing from Illumina, where it's called VeriSeq. You can also see the similar level of sensitivity for detecting aneuploidy in a known level within these cells. But what people have started to do is increase their sensitivity to predicting these abnormalities by essentially lowering thresholds. So instead of requiring that the copy number reaches 2.5 or 2.6, you might lower the threshold so that if the copy number for a chromosome is only 2.3, you may actually predict that it's abnormal. And by doing that, and this custom VeriSeq is essentially just changing thresholds for predicting aneuploidy, you can actually increase the sensitivity and start to detect these things earlier on. And this is what many groups have done in order to claim their ability to detect mosaicism using next-generation sequencing. But I think one of the things that we need to consider is then by changing these thresholds, how does it impact false positives? So do we start detecting things that we shouldn't? In this example, when you use custom thresholds, so you make the test more sensitive, you start to predict mosaicism for chromosomes, which we know in this particular sample are not mosaic. This is actually uniformly euploid for chromosome 2, but because you have this shift in the copy number, it's going to be called mosaic. And here you see that the decrease in specificity is associated with this increased sensitivity. So you now introduce more false positives and start to call more embryos mosaic when they actually aren't mosaic. And many of you have probably seen this study published in the New England Journal of Medicine, which suggested that healthy babies can be born from mosaic aneuploid blastocysts. And I want to point out really the lack of evaluation of specificity and false positives in any of the control data. and potentially an alternative explanation for the results. So here they found that about 1/3 of the embryos that they predicted to have mosaicism led to healthy babies. And I would suggest that most likely in this situation, those embryos were never mosaic to begin with. And the test actually produced a false positive, which is why these embryos were able to develop into healthy babies. And I also want to point out, so and a lot of people have argued that the validation of predicting mosaicism is based on the fact that these so-called mosaic embryos have a lower reproductive potential. And I think there are many other ways to validate the technology. I showed you the cell line mixture model. You could also reevaluate embryos predicted to be mosaic to see if there's actually mosaicism in the remaining embryo. But most people have just suggested it works because you have a lower success rate with these embryos. And I think one interpretation is that perhaps in some cases, the test is just failing to identify uniform aneuploidy in these samples. So it's what I refer to as a false negative uniform aneuploidy. Instead of calling it uniform aneuploidy, for whatever reason, the signal for the aneuploidy is decreased and falls in the mosaic range, when it actually should fall in the full aneuploid range. And I'll show you some data on that. In this example, the first biopsy indicating a trisomy of chromosome 13 might fall into this mosaic range, and you would predict that embryo to be mosaic. But upon rebiopsy, you may find uniform trisomy of chromosome 13. And in a recent presentation by the group at CCRM, they actually found a relatively low mosaicism rate overall, which means that they've set their thing. But among those 3% predicted to be mosaic, 1/3, when they performed rebiopsies, were actually found to have uniform aneuploidy. So again, it's a false negative uniform aneuploidy. So some embryos being diagnosed as mosaic are actually uniformly aneuploid. And I'm sorry, I'm going to talk a little bit about molecular biology. And this is just illustrating how you might observe intermediate copy number when you should see a full trisomy. And it has to do with amplification kinetics. You can see this sigmoidal curve. So when you're amplifying DNA, you can test the quantity over multiple cycles of amplification, and you typically see this exponential curve. And what you'd like to do is evaluate the sample in the linear range so that every cycle there's a twofold increase in the quantity, which just means you're able to double the amount of DNA after each cycle. If you were to test, and for example here, if you tested the sample at this point, so you performed, let's say, 25 cycles of amplification, you would see a larger difference between trisomy and disomy. The disomy chromosome amplifies later because there's less DNA. The trisomy chromosome amplifies sooner because there's more DNA. But once you get into this saturation where there's really not enough reagents to double the amount of material at each cycle, and so you can't continue to increase, this starts to flatten out. And if you were to test the sample, let's say at 30 cycles, that difference between trisomy and disomy would be smaller. And that's why you might see these intermediate copy number values because there's saturation. Hopefully that makes some sense. And there are a variety of other phenomena that can contribute to these observations of intermediate copy numbers that aren't actually due to mosaicism. An example is contamination, and this isn't something that has been well evaluated in aneuploidy screening. It's typically been evaluated for single gene disorders where contamination can be a big issue, and it's really been overlooked with aneuploidy screening. There are all kinds of sources of aneuploidy, sperm, cumulus cells, DNA in the media, technicians, sibling embryos that might have remnants when you perform multiple biopsies within a cohort. And most technologies that perform aneuploidy screening don't have genotyping capability, which would actually allow you to look at contamination. In this situation, you're just looking at copy number, and so you might consider that this is a mosaic for chromosome 17, just based on copy number in both of these two platforms for next-gen sequencing. But when you have the ability to genotype, you can actually distinguish between what would be a pure sample, where you expect, for example, positions in the genome that are heterozygous. You have a 50-50 ratio of the two alleles. And so many of these SNPs will cluster around 50-50. When you have a contaminated sample, they become all over the place, so you can actually identify contamination. And in fact, this was a sample that we artificially mixed to different individuals' cells, so we know that they were contaminated, and you could detect it with genotyping. So in this situation, the mosaic range aneuploidy call is not due to mosaicism, it's due to contamination. Another phenomenon, and I think we've estimated that about 1 in 200 embryos has triploidy. In this situation, it was not a full triploidy, so there was 3 copies of all of the chromosomes with the exception of 2. And if you just had copy number, you would consider this to be a mosaic monosomy for chromosome 2. But when you look at the allele ratios, you can see that instead of 50-50, they cluster around 1/3 and 2/3 because you have 3 copies of each allele, or each chromosome, so you can have 2 of one allele and 1 of the other, rather than 1 of each. And so this allele ratio analysis on the genotypes can allow you to distinguish between, in this case, a mosaic embryo and an actual partial triploid embryo. And this might be considered for transfer at some places because it's a monosomy and chromosome 2, when in fact it has no reproductive potential. So, to conclude, I think mosaicism is overestimated by inaccurate methods and weak criteria. Intermediate copy number can originate from phenomena other than mosaicism. And I think criteria for diagnosing mosaicism from a single biopsy needs evaluation of not only sensitivity, but also specificity to acknowledge that there are false positives. And because of that, I think that it's more responsible to, if in fact you're going to try and detect this, to report the results as consistent with possible mosaicism and not a definitive diagnosis of mosaicism. And then to answer the question, I think the point is, don't use a test which claims to diagnose mosaicism from a single biopsy. Thank you.
Thank you, Nathan. I think I would like to use the chairman privileges here and ask you a couple of questions before opening to the floor. So the first thing that I would like to say is actually not a question, it's a comment about the fact that screening for aneuploidies in general, not mosaicism, improves results. And I hope I'm not jumping the gun on George's talk, but I just would like to point out that like any other selection or analysis that we do on embryos, we are not really improving them, we are just ranking them. So for this reason, the cumulative pregnancy rate should be the same. And since we are not really treating embryos, we are treating patients, I think at best what we can do is to decrease the time until we get a live birth. But in order to know if we do that, we should see if PGS improves, starting from the people that enter the process at stimulation, not from the transfers, because we all know that once you get to transfer, those that get to transfer, they will have better outcome in that transfer. That's just a general comment.
Sure. Do you mind if I reply?
Of course not.
Okay. No, I completely understand that we're not changing the status of the embryos. Obviously, the patients drop out after multiple attempts, and obviously miscarriage will delay the potential for a subsequent cycle. Cumulative outcomes are important to understand, but most patients would prefer to get pregnant on the first attempt, and there are well-documented issues with dropping out after multiple cycles. So I'm not arguing that we're changing anything, but I think selection is important to improve success rates on the first attempt.
I absolutely agree that selection is important. I just want to point out that its improvement should be measured on patient, not on transfer. That's
and I've heard that argument before, and one of the randomized controlled trials was actually intention to treat, which I think people fail to acknowledge
Yeah.
that on an intention to treat basis, there was an improved outcome on the first.
Yeah, that's the reason why, for instance, we do use PGS in very good prognosis patients, because we think they're the ones that stand to wait longer to have a live birth. But any case,
thanks.
My question for the mosaic embryo is, so I realize, so the suggestion is laboratories should not report embryos as mosaic, but with the makeup that is suggesting of mosaicism. Okay, then we get these reports, and what do we do?
Well, as soon as you start getting those reports, request that you no longer get those reports.
Okay, so
and
we
unfortunately
should not
you have,
want to know, basically.
Yeah, now you're in the situation where you have all of these embryos predicted as mosaic. And I think it's an unfortunate situation. I don't have the answer to how to deal with those, but I do think that going forward you might want to reconsider. And I do think many groups now performing testing do offer the option of not reporting prediction of mosaicism, and so I would suggest doing that. So the data I showed where we developed controls for known mixture levels of aneuploidy, I think it would be more responsible to just provide that information to patients so that they know that, okay, for example, maybe 5–6% of embryos do have mosaicism, and amongst those, if there's greater than 50% mosaicism in the biopsy, we're going to detect it and call it abnormal 40% of the time. So provide that information rather than making a definitive diagnosis of mosaicism, but rather describing the test capabilities and limitations, so when they get an abnormal result, they know what it means.
There was a question from the audience.
Thank you very much for your talk. It was very important and interesting. For long years, we've been told that PGS 1.0 was very efficient, and then it was proved to be not the case. Now we have PGS 2.0 that was proposed as the silver bullet in order to determine aneuploidy. But now you're telling us that there are a number of different methodologies that are flawed. So I'm just wondering how much we need to wait to have a proper technology which is accepted on a consensus by everyone.
Yeah, I mean, this industry is a lot like the mafia. I think you're not going to have a consensus ever because there's commercial interest. I think that, unfortunately, part of the issue around mosaicism was based on companies wanting to have commercial advantage and suggesting that they can detect mosaicism better than anyone else. But it's also not as simple as bulking everything into PGS 1.0 and PGS 2.0. I think it's easy to oversimplify. And we've seen, even with FISH technology, when applied in a specific population with a very good laboratory, there is a randomized controlled trial showing that it does improve outcomes. And then with PGS 2.0, there are unfortunately a variety of technologies with different levels of validation. But there are some platforms which have gone through rigorous validation, including preclinical analysis of positive controls, establishing performance on those controls, as well as randomized controlled trials showing benefit in certain populations. So it's not that there isn't access to these technologies. I think it just requires people to invest a little bit more time in understanding what is available and what the evidence is to support its particular use over other platforms of PGS.